pasado 4 de octubre, desafortunadamente tuvimos el sensible fallecimiento del doctor Héctor Mayagoitia Domínguez, quien da nombre a esta cátedra. Mandamos nuestras condolencias a la familia, amigos, y nosotros continuaremos con los trabajos del presente evento, poniendo en alto su nombre como un destacado y distinguido politécnico. Por lo que en el marco de la Cátedra Patrimonial en Medio Ambiente, doctor Héctor Mayagoitia Domínguez, octava edición, versión 2023, tenemos el honor y es un placer para mí en presentar a la doctora Luo con la conferencia titulada Plasmonic nanotools derived from all diazonium salts for bioimaging and sensing. Doctora Lu, I'm going to read your biography in Spanish. When I finish, I will say, Doctora Lu, you can start. ¿Ok?
Okay.
Ok. Daré una pequeña semblanza de introducción de la trayectoria de trabajo de la doctora Lu para que ella esté atenta en qué momento compartir su presentación. Bien. La doctora Lu recibió su doctorado en química de materiales en el Instituto Nacional de Ciencias Aplicadas en Rennes, Francia, por sus siglas en francés, INSA, en el 2012. Luego ganó su primer puesto postdoctoral en la Universidad de Rennes I, donde participó en fotosíntesis artificial. Posteriormente, se unió al equipo del profesor Nicolás Alonso Bante en la Universidad de Poitiers como postdoctorante en el proyecto de investigación europeo sobre pilas de combustible, principalmente estudiando la reacción de reducción de oxígeno en el cátodo. En el 2016, la doctora Luo fue nombrada profesora asociada en la Universidad Tecnológica del Sur de China, desarrollando nanomateriales avanzados para la conversión de energía. Continuando con su trayectoria académica, ocupó un puesto de profesora e investigadora asociada, hace por sus siglas en francés, en la Universidad de París Descartes, París 5, del 2017 al 2019, centrándose en la funcionalización de nanomateriales plasmónicos por espectroscopía Raman. En el 2019 obtuvo un puesto de científica investigadora titular, CRCN por sus siglas en francés, en el Centro Nacional de Científicos Investigadores, CNRS por sus siglas en francés. Actualmente dirige el equipo de nanobioespectroscopía en el laboratorio LCBT CNRS de la Universidad París-Cité desde el 2022, donde impulsa la investigación sobre la funcionalización de superficies de biomateriales, con especial atención en la bioimagen multimodal, suministro multifuncional de fármacos, diagnóstico, terapia del cáncer e implantes de dispositivos médicos, con el objetivo de mejorar las tecnologías médicas y la calidad de vida de los seres humanos. La doctora Lua es coautora de más de 40 publicaciones revisada por pares y recibió 2 proyectos de la Agencia Nacional de Investigación de Francia, ANR por sus siglas en francés. Sin más preámbulos, without more introduction, we leave the spotlight to Dr. Alu, where she can start to present her presentation. So we have 40 minutes, Doctora, for your presentation and 15 minutes for question. Okay? You can
Okay.
start to share your presentation, Doctora.
Okay. So thank you, Luis, for your invitation and organization of this seminar. It's really a great pleasure to be here to introduce you the latest work of our team on plasmonic nanotools derived from aryl diazonium salts for bioimaging and biosensing. Maybe you have already asked a question, what's plasmonic nanoparticle? As you can see from this figure, the plasmonic nanoparticles on the light irradiation, the surface free electron we call electron cloud start to oscillate, and such a movement will generate electric and magnetic field. This phenomenon we call localized surface plasmon resonance. And such a phenomenon gave plasmonic nanoparticles excellent optical property. The first interesting property is the strong absorption of light in visible region. As you can see from this photo, there is a plasmonic nanoparticle suspension, and the concentration of this solution is down to maybe nanomolar. However, you can see it has very strong and intense color. This is because plasmonic nanoparticles can have extinction coefficient million times greater than molecular chromophores. And such absorption can be extended to near-infrared region if we change nanoparticles type, size, or shape. The second interesting property is plasmon resonance is highly depending on medium or environment. As you can see from this figure, the plasmon band of nanoparticles can be shifted if we add a drop of oil into the aqueous solution. And such a shift is proportional related to the oil concentration. So we can see plasmon nanoparticles are very sensitive to the surrounding medium. This property can be used for the detection. The third interesting property is giant amplification of Raman spectra of an analyte, usually a molecular, adsorbed on nanoparticle surface. And such an effect we called surface-enhanced Raman spectroscopy, SERS, which can boost detection limit down to single molecule level. So nowadays, plasmon nanoparticles is becoming very popular because it can providing novel nanotools for various applications such as nanosensor, meshing, photocatalysis, and so on. For all those applications, it will require to control the surface chemistry because the surface is the key parameter determining the nanoparticles interacting with surrounding environment. To control or functionalize plasmon nanoparticles, usually we use a coupling agent we call thiols. This kind of molecular result thiolate surface sampling monolayer. As you can see here, it's a homogeneous and compact monolayer. However, it suffers from several limitations such as desorption and thermal instability. So in our group, we propose to use another coupling agent called aryl salts, as you can see here, because it can create more stable covalent bond between carbon and metal. And if we change the position of functional group to para or ortho position, this will lead to monolayer or polyaryl multilayers. And of course, we can select it appropriate functional group to adapt our applications. For example, if we want to use nanoparticles for Raman imaging, we can select nitro or cyano group, which can serve as Raman tag. If we want to apply nanoparticles for targeting, we can select carboxylic acid, which allow post-functionalization of biomolecules like peptides or proteins. In fact, based on our previous results, we have proven aryl salts are very efficient to functionalize, for example, aluminum nanoparticle surface, gold nanorods, silica, gold island, or nanostrip propelled by lithography. In fact, the application of aryl salts have been already used for biosensing, bioimaging, target therapy, drug delivery, and antibacterial application. But the use of aryl salts for the design of search tag based on plasmon nanoparticles has never been reported before our work. So the whole project is trying to combine plasmon nanoparticles with aryl salts to design a new generation of SERS encoded nanotools for Raman imaging and Raman sensing. The results I'm going to show you are all extracted from the Ph.D. thesis of Dali and Yu Xiao. Let's start it with the design of search tag for cell multiplex bioimaging. In the domain of optic imaging, we always ask a question, how can we increase the information content? As you can see from this photo, there are only two colors, the pale yellow and black one. So for me, for this baby zebrafish, I can only distinguish eyes from the body. If you ask me where to find bone, skin, stomach, I have no idea. But if we can stand them and we can maybe see them. So the idea is very simple. If we can add more color, we can add more information. In fact, such a philosophy has been used in our daily life. For example, if you are traveling in Paris, you will get this map. There are 14 metro and 5 RER line, and each line is color unique. Because this will help you to distinguish one line from the other one, and that will help you to get your destination successfully. In the domain of bioimaging, the most common used way is three-color fluorescence imaging. As you can see this photo obtained on the fluorescent microscope, we stain the nuclear in blue, protein in green, and membrane in red. But if you want to have more details, for example, where to find liposome, DNA, we need to go beyond conventional three-color bioimaging. Nowadays, we can use genetic engineering to realize multiplex bioimaging. As you can see from this photo, this zebrafish, each cell on its skin can express a unique combination of green, red, and blue fluorescent proteins, resulting in over 17 different hues that can be distinguished under a microscope. This helps researchers study how skin regenerates and heals. However, such a technology requires genetic engineering. It's expensive and time-costing, and I'm not sure if we can do this for every patient in clinic who just want to do a diagnosis of disease. So we need to find a simple and low-cost way. Nowadays, we can do multiplex bioimaging using fluorescent tags based on quantum dots. We can also use search tag based on plasmonic nanoparticles. The search tag is composed by surface-confined Raman reporter, usually a molecule, onto plasmonic nanoparticle. Compared with fluorescent dye, search tag has several advantages. First of all, compared with a single peak of emission, Raman can give multiple sets of narrow peaks, and each peak corresponding to a mode of fabrication of a chemical bond. So in addition to optic information, Raman can give structure information that might be very useful to understand how nanoparticles interact with biologic surrounding. If we want to realize multiple fluorescent imaging, we need more emission, and more emission might require more excitation. But for Raman, single light source can be used to excite multiple Raman reporters and with negligible photobleaching. And for the Raman, we can also excite the signals using near-infrared light source. These fibers deep tissue analysis in clinic. Finally, compared to quantum dots, plasmonic nanoparticles has relatively low toxic, which is very attractive for in vitro and in vivo bioimaging or sensing. The only difficult is how to obtain stable nanoparticles with various Raman reporters. So in our project, we propose to use plasmonic silver or gold nanoparticles functionalized by aryl diazonium salts. First of all, we have synthesized the colloids by mixing the silver and gold gold precursor with reducer, and we also add some polymers to stabilize nanoparticles. And then we mix the colloids with a solution containing aryl diazonium salts for 1 hour. As you can see after the grafting, we can visualize a layer of polyarea on the surface of nanoparticles, indicating molecules has been grafted on nanoparticle surface. To obtain multiple search tags, we propose to use a step-by-step strategy. That means we incubate nanoparticles successively with aryl salts but bearing different functional group, which can be served as different Raman tags. It is worth noting that during this process, nanoparticles stay very stable. As you can see, the plasmon band has been slightly shifted. This is because the surface chemistry has been changed after the grafting. However, the band shape is almost the same, and it's very symmetric. This indicates there is no agglomeration formed during the process. And then we characterize source tag using Raman spectroscopy. For the nanoparticles derived from aryl salts bearing CC alkyne group, and we can detect the CC peak in its spectrum. For the nanoparticles bearing NO2, a neutral group, we can detect this peak in the spectrum. For the nanoparticle functionalized by aryl salts bearing CN cyano group, and we can detect this peak in its spectrum. And for the nanoparticles co-functionalized by aryl salts bearing NO2, CN, and CC group, and of course, we can detect NO2, CC, and CN peak in the Raman spectrum. So these results showing successfully grafting of aryl salts on silver nanoparticle surface, and which can be used as source tag. If we define the Raman signal of NO2 peak in red color, that of CC peak in green color, that of CN group in green color, and according to RGB color mixing principle, we should have a mixed color for the nanoparticles co-functionalized by those three types of aryl salts. In order to check, we have incubated HeLa cells with those four types of nanoparticles. As you can see here, we can only see the cells in red channel if it's incubated with nanoparticles bearing NO2 group. Only in green channel if it's incubated with nanoparticles bearing CC group. And only in blue channel if it's incubated with nanoparticles bearing CN group. And in red, green, and blue channel for the nanoparticles bearing NO2, CC, and CN group. So this is a proof of concept for promising prospect for multiplex Raman bioimaging using source tags based on silver nanoparticles. Here we have checked the cell cytotoxicity of the source tag based on silver nanoparticles using Alma blue arrays. As you can see, with the nanoparticle concentration up to 20 microgram per milliliter, the cell almost dead if it's incubated with nanoparticles without any modification. However, the cell viability can be increased if it's incubated with nanoparticles with modification. So this suggests surface functionalization by aryl salts tend to reduce cytotoxicity. We have also developed source tag based on gold nanoparticles using the same strategy. As you can see here, those nanoparticles can be also uptaken by cells and also can be used for bioimaging. However, the staining process, the incubation time is much longer, up to 24 hours. Concerning to cytotoxicity, as you can see, when nanoparticle concentration up to 200 microgram per milliliter, we didn't observe obvious toxicity. So we can see maybe for bioimaging, gold nanoparticles is safer than silver nanoparticles. So in the second section, we were wondering, okay, silver nanoparticles is a little bit toxic, but why not use them, apply them out of biological domain, for example, in optical imaging for anti-counterfeiting. Anti-counterfeiting is research area in priority for social economy and public safety, because the global economic value of counterfeiting could reach $4.2 trillion by 2024. This technology requires hiding information from public, but can be identified under specific condition. For example, if you want to know a cache is true or fake, you can put it under the UV light, and if there are some fluorescent patterns tags appears, that means the cache is true. So as you can see, currently anti-counterfeiting technology is based on fluorescent labels, which suffer from emission peak overlap, photo bleaching, and it's easy to be counterfeited. So we propose to use search tag because search tag, we encoded information. The information we encoded is the Raman peak, and the Raman peak is related to structure information of the tag. And we change them to color, we transfer them to color, which is not easy to be counterfeited. Similar to optic imaging, if we want to hide more information or if we want to encode more information, we need more color. So we propose to use silver nanoparticles functionalized by aryl diazonium salts bearing CC, CN, and NO2 group as 3 primary colors, and then mix them 2 by 2 or 3 of them to generate more colors. In fact, the ice-propelled silver nanoparticles, even after the functionalization by aryl diazonium salts, cannot be directly used to fabricate anti-counterfeiting tags labels. And we need to disperse them in a very special ink, which has appropriate surface tension and viscosity. And then we can use this ink to fabricate anti-counterfeiting label. The first thing we need to do is to check the source signal. As you can see, the signal is almost unchanged after dispersion into the ink, indicating nanoparticles is very stable. And then we loaded one ink in a cartridge and loaded in a pen. As you can see from this photo, we have written a very beautiful poetry from Hemingway. And of course, from leaky eye, you have no information which ink I have used. This information can be only reviewed under a Raman microscope. So now we can see we have used an ink containing silver nanoparticles bearing NO2 group to write down the word of Paris. Let's go further. Here we have write down the name of our laboratory. And of course, you have no information which kind of ink I have used to write down the name. But thanks to Raman microscope, we can see for the letter U, we use the ink bearing NO2 group. For the letter M, we use the ink bearing CC group. And R use the ink bearing CN group. 8 use a mixture of NO2 and CC ink. 6 the mixture of CC CN ink. 0 mixture of NO2 CN ink, and 1 the mixture of 3 ink. Here we have investigated the stability of our source ink. By treated our handwriting use different liquid, water, ethanol, oil, acid, and alkaline solution. As you can see the handwriting after the treatment of water and ethanol, the signal of source is almost no change. After the treatment of oil, acid, and alkaline solution, the characteristic peak intensity reduced from 25 to 10%. Here we have compared the stability of the source ink derived from aryl diazonium salts and conventional Ziols coupling agent, and we treated them in acid and alkaline solution. As you can see, for our homemade ink derived from aryl diazonium salts, the peak intensity has been slightly reduced after the treatment. However, we can observe a dramatic decrease of peak intensity if we use the ink derived from Ziols. For the photostability, we observe the same tendency. For the ink derived from aryl diazonium salts, under laser exposure for 10 minutes, not too much change we can observe. However, for the ink derived from xylose, there are new peaks appears. This can be related to the plasmon-driven formation of azobenzene. So those results show the ink derived from aryl salts is much more stable than the ink derived from conventional xylose coupling agent. At the end of this project, we try to apply our ink for inkjet printing because this technique is no contact digital deposition that can be easily adapted to any size or production scale, from small substrate to large scale, large area industrial production. So we fill the inkjet printer with one ink and the print of university logo, as you can see here. And of course, from eyes, you have no idea which kind of ink we have used, but we can use Raman spectroscopy. We use the ink containing CC group. It is worth noting that inkjet printing have already used to fabricate anti-counterfeiting tags. However, the conventional way is always two-step. The first step, we deposit plasmonic nanoparticles. And the second step, we deposit Raman tags. So, to the best knowledge, our work is the first example of direct inkjet printing of all-in-one surface-active nanotags. In the last two sections, we have explored a surface tag based on silver and gold nanoparticles for Raman imaging. And in this section, I'm going to show you some preliminary results on surface-modified silver nanoparticles for Raman sensing. As proof of concept, we focus on a very important Raman sensing topic, glucose detection for the diagnosis of diabetes. Diabetes is a disease affecting 4 million people in worldwide. The standard detection is based on high-performance liquid chromatography or enzymatic approaches by analyzing the glucose level concentration in blood. Such a technique is invasive and very expensive and of course time-taking. So researchers are trying to find a simple and non-invasive way that is detection of glucose in urine. However, since long time, this technology suffers from poor limit of detection. As you remember at the very beginning of my presentation, I've said a search technique can boost the detection limit of analyte low to single molecule level. So search technology is a very interesting and ideal approach to improve the detection limit and sensitivity for glucose detection in urine. The mechanism for glucose detection in urine is based on boronic acid receptor. Because this molecule has high bonding affinity with monosaccharides. And among all the sugars, only the glucose can form bidentate complex. So the formation of such a sandwich-like structure can be only in presence of glucose. The conventional fabrication of surface sensor is shown here. We need to firstly graft a receptor on nanoparticle surface, and then incubate the sensor in a solution containing glucose, followed by probe molecules. The probe molecules containing boronic acid as receptor to capture the glucose. It bearing also a Raman tag, so we just need to follow the appearance of this Raman signal of the tag to know if in the solution we have glucose or not. Such a method is high sensitive and no interference. However, it suffers from no reproducibility and it's hard to be used for source quantification. So in our work, we propose to use a second probe, which is directly immobilized on silver nanoparticle surface. The first probe we can use to review if there is glucose or not. And the second probe is internal references to calibrate source spectral fluctuation. And the ratio of peak intensity of the first and second probe, we can use them to improve the accuracy of quantification of glucose. In our project, we go further. To facilitate and simplify the sensing process, we have fabricated a sensor chip. By immobilization of silver nanosensor onto planar 2D iron surface. And we put this chip in a solution of glucose, followed by probe molecules, and submit directly under the Raman microscope for detection and analysis. Here is a follow-up of the preparation of sensor chip. As you can see, before any treatment, according to Raman or ACM and EDX, there is only iron can be detected on iron surface. And then we use iridadium salts bearing CC group, which serving as anchoring sites for silver nanoparticle and also internal references. And as you can see, after the grafting, the peak of aryl appears, indicating we have grafted these molecules onto iron surface. And then we incubate the surface modified iron with silver nanoparticles pre-functionalized by iridadium salts bearing boronic acid receptor. As you can see from the spectral and ACM, we can find the pre-functionalized nanoparticles has been successfully immobilized on substrate surface. And then we use source mapping technology to check the uniformity of source signal on the chip. As you can see, the signal is quite homogeneous and stay very stable over 3 weeks under air and in room temperature. And finally, we incubate the chip with a solution containing different concentration of glucose, followed by incubation with probe molecules. From the Raman spectral, we can clearly observe the internal references peak. For the probe molecular peak, we can find the intensity can increase with increasing of glucose concentration. And such a phenomenon can now be detected if we replace glucose to fructose. So this indicates our sensing system has very good selectivity. And finally, we use radiometric analysis to check the peak intensity versus peak intensity of probe versus references. And our system has a limit of detection as low as 2.6 micromolar. Here we have validated our chip for analysis glucose in artificial urine. The final concentration we have found in the urine is 2.5 millimolar glucose, which is perfectly match the theoretic concentration that we added in the artificial urine. Here we have compared our sensing system with that published in literature. As you can see, our system has lowest limit of detection and widest linear range for glucose detection. This means our system is superior compared with other systems based on source. Same mechanism. Conclusion and perspective. In summary, we have associated plasmonic nanoparticles with iridadium salts to develop innovative nanoscale tools for Raman imaging and sensing. In the first section, we have fabricated silver and gold nanoparticles derived from iridadium salts as source tag for multiplex Raman bioimaging of cancer cell. In the second section, we have developed new kind of source ink for for handwriting and inkjet printing as anti-counterfeiting targets. And in the third section, we have developed diazonium multiphased chip sensor for sensitive and selective glucose detection. For perspectives, for the first section, we would like to develop new insights on biodegradation of nanoparticles once uptaken by cells, and their long-term stability in complex biologic environment. For the second and third project, we propose to strengthen the cooperation with the field of computer science, microelectronics, and mathematics to improve the development and practical application of 7 nanoparticle-based search sensor and anti-counterfeiting targets. For all the projects, we hope to explore prospects of diazonium functionalized plasmonic nanotools and extend their applications in another domain such as energy, catalysis, and environment. And we hope to establish coordinated and collaborative investigation covering chemical, biology, toxicological, and industrial aspects to boost the transfer of these laboratory products to real-life application. At the end of my presentation, I would like to shortly introduce our university. Our university is called Paris City University because all our campus can be located in Paris City. And we are quite new because we are just created in 2019 as a fusion of Paris Descartes University, Dito University, Paris Global Physics Institute, and now Pasteur Institute is joining us. Our laboratory is located in the heart of Paris. As you can see, it's now very far from Musée du Louvre. And the research topic for our laboratory is on the interface of biochemistry, chemistry, nanotechnology, and biology. Our research team, nanobiospectroscopy, has 4 permanent staffs, 1 PhD student, 1 postdoc, and each year we will host 2 to 5 master students. And the research topic for our research team is focused on surface functionalization of nanoscale, microscale, massive materials for biomedical applications. And of course, we're developing also new method approaches for Raman imaging and sensing. At last, I would like to acknowledge all the team members, all our collaborators, and you. Thank you for your listening.
Okay. Muchas gracias, doctora. Thank you so much for your presentation. It's a nice and interesting topic related to the healthcare, right? So, es muy interesante la parte que nos acaba de mostrar la doctora, porque esto desarrolla sensores a nivel de aplicación que pueden no ser invasivos, ¿no? En el caso de que ella está detectando glucosa a través de la orina. Vamos a verificar si hay algunas preguntas. We have to check if there is some question, or there are some questions, sorry, there are some questions on the YouTube channel.
Okay.
Okay. Okay. Yes, we have one del el doctor Juan Alberto Alcántara Cárdenas de aquí del CIEMAD. He asked Dr. Alberto Alcántara how to improve the stability of nanoparticles plasmonic in biological environments. Do you have some ideas about that?
Actually, if you want to stabilize nanoparticles before cell uptaking, you can use some polymers to stabilize or some capping molecule to stabilize. But once it uptaken by cells, it start to aggregate. So actually, as you can see, the Raman imaging, all you see is not a single nanoparticles. They are agglomerations. So they are tend to agglomerate in cells.
Okay, nice. We have to check another one. We have time. Um, okay. You said about that you have to collaborate with persons who works in electronics, right?
Yes.
And do you have some ideas about how to connect these sensors with the electronic, for example, with Arduino, with another related topic? Do you have some ideas about how to manage that?
Yeah, actually, we are looking for a collaborator because we want to combine our sensors to, for example, computer science to mainly analyze the data. For example, we want to use artificial intelligence to
Mm,
check, yeah, to analyze all the data, all the spectra we have obtained. Because now with our naked eye, if there's more differences, maybe it's related to different phase of disease or different type of disease. And from our eyes, it's not easy to be distinguished. But I think it's not a challenge for artificial intelligence. So that's a mind domain we want to extend it.
oh, right. Related to the cancer, it's possible to detect in a good time before you have this disease?
I'm sorry, I don't understand the question.
Okay, okay, no problem. If you detect cancer, how many time or in which time you can prefer to analyze in order to tell the person if that's okay or it's not cancer, you know.
Okay, so so if we use the Raman technology to analyze to diagnosis of cancer, the Raman analysis is very quick.
Mm.
So a spectral can be obtained in seconds.
Mm.
But there are maybe some before the detection by Raman, maybe there are some you need some time to sample preparation. That
Mhm.
might take, I would say, it's a classic procedure. So the
Okay.
most time saving is in the face of Raman detection.
Oh, okay, okay. So for the doctors or medical doctors, they will have in seconds or they reduce the time to detect that cancer, right? So,
Yeah.
wow. So, es interesante porque podrían detectar si la persona tiene cáncer o no. Y justamente eso daría mucho tiempo de respuesta para el médico poder responder al tratamiento, ¿no? Okay, there is another question we have here in YouTube. Dr. Raciel Jaime Lopez from CIEMAD. Thank you very much. Could you please summarize what characteristic of nanoparticles must have to have an adequate plasmonic response?
I didn't get this question. Do you want to ask what kind of nanoparticles is the best for plasmonic analysis? Is that
They say that is which characteristic in nanoparticles must have to have
to
in a
check?
plasmonic response. I think if we change
Yeah.
different nanoparticles that had a plasmonic characteristic or behavior, which
Yes.
the adequate response for that.
Uh, well, it's if I will understand your question, you are asking which kind of nanoparticles is most adapting the analysis, right?
Yeah, yeah.
Okay, so it depends on the application. If for a clinical applications, I will say the most adapted nanoparticles maybe should have excitation in near infrared region,
Mm,
because in near infrared region excitation, the beam can penetrate from the tissues, so you can have deep tissue and like a deep tissue response. So in this case, we need to well-selected nanoparticles because you say nanoparticle type, shape, and size might change the plasmon band, the absorption. property. So in this case, I propose to use gold nanorods because gold nanorods is like this. Well, it has two mode of two mode of plasmon band. So it can be excited in near infrared region. So it's a good idea. And also nanorods, nanorods maybe can pass the filtrations, the infiltrations of the kidney, so it can be a very quickly degradation.
Oh, okay. So it depends on the morphology of the nanoparticles, right?
Yes.
Oh, right. Okay, okay. Well, we have no more question. If another person o alguna otra persona, perdón, pudiera
Mhm.
hacer alguna pregunta, podemos aquí comentarlo. Todavía tenemos algo de tiempo. Solamente pueden colocar su nombre, dependencia o institución y la pregunta, por favor, para aquellos que deseen preguntar, que todavía estamos en tiempo. Podríamos dar algunos segundos si a alguien más le gustaría preguntar. Por ahí también se les compartió el registro del resumen de la conferencia para que puedan obtener su constancia de asistencia, y esto es para los estudiantes que han aplicado, por ejemplo, a becas BIFIN. Ahí pueden enviar su resumen y podrán tener justamente una constancia de haber estado presentes en la conferencia. Ok, there is no more question, doctora, about the topic. It's very interesting topic. We can here talk about a lot of time. And how to manage this related topic with electrochemistry. I think it's so interesting for everywhere and everyone, right?
Yeah.
Ok, pues yo creo que puedo compartir la, la, prese- la puedo compartir el certificado. Thank you for your questions. I'm going to read and share the certification. Muchas gracias por sus preguntas. Ahora continuaré con mostrar el diploma de la doctora Lu. I will share your certification. I will read, Professor. Certificate for Ph.D. Jun Luo for the keynote lecture, Plasmonic
Oh,
Nanotools
thank you.
derived from aryl diazonium salt for bioimaging and sensing. Cátedra Patrimonial en Medio Ambiente, Dr. Héctor Mayagoitia Domínguez. Excellence Professorship in Environmental Science. Mexico City, 17 October 2023. La técnica al servicio de la patria. Bien, the diploma is singing by the principal of here, nuestro doctor Pedro Francisco Rodríguez Espinosa, director del CIEMAD, firma el certificado. I will share you that in later, Professor John. I will share you later the certificate in order to do you have that one. Okay,
Okay. Thank you so
okay.
much.
If there is no more
Great honor.
questions, si no hay más preguntas, si no hay más comentarios, pues podríamos dejar prácticamente cerrada la presentación y podríamos esperar la siguiente presentación, que más adelante se les podrá indicar en el mismo canal de YouTube cuál será la presentación del siguiente ponente y el nombre del ponente. ¿Ok?
Okay.
Mm.